Today, I'm going to talk a little bit about the discussion. One thing to know about the discussion is this is worth a big chunk of your report grade. So I think it's 40% of the full grade in terms of all the sections. This is going to be the longest section. I would say this is a minimum of 2 pages, maybe 3 pages. And what you're doing is you're discussing, interpreting, and talking about the results. So remember in the results section, what you are doing is basically presenting them. Here they are, here's the numbers, here's the graphs, right? The discussions section is where you're interpreting them. You're saying, okay, this is what the results actually mean. And to guide you through that, I have a bunch of questions in the lab manual. And if you answer all those questions, you're probably in pretty good shape. Like I said, always good to give a little bit more. So just remember to be specific, complete, and thorough. And don't just give me answers off the top of your head. I don't want the grade 9, grade 10 answers. I want you to go to your class notes. I want you to spend a little time on Google making sure that you're giving me nice, good answers on this. Here is an example, or 2 examples. And you can see that the first one versus the second one. In fact, the first one is not bad, but in my mind, it's a good start, right? It's not telling me anything specific. It's telling me something there about freezing, okay, is damaging, that's good. But what you need to do now is go on and prove it. Prove it by discussing the data. So this is telling me that freezing, well, what does that mean? In this case, we tested -20, okay? And it's telling me exactly how much damage we did. And it's saying, if you don't believe me, you know, even go back and look at figure 2 and you can see all of the data all in place. So I really like that one. It's very specific, it's concise, it's telling me some good information. Like I said, that's not necessarily going to be enough. You need to interpret this, tell me why. Why is it that freezing temperatures are more damaging to membranes? Like, why would that be? And even better is to give me an example of a study that has done exactly that, to confirm that your findings are consistent with what is found in the scientific community. Now, in Biology 107, I'm not expecting you to do a bunch of intensive research where you're looking up journal articles and things like that. Remember, you need one journal article that you're going to be referencing somewhere. And mostly I want you to show me that you can cite it somewhere, reference it somehow. And you can do that in the introduction section, or you can do it in the discussion section. As you get further on in your education, it's going to be expected, though, that you do a lot more research. And it kind of gives authority to answering your questions. It's not just you finding that information, but there is consensus in the scientific community. It gives a lot of authority and weight to what you're finding. I'm not going to say too much more about the discussion, with the exception of one thing. Okay, something that a lot of people get confused about is what is going on with these solvents. So take a look at these solvents. Take a look at the chemical structures of them. We use water, methanol, ethanol, and propanol. What do you see that's different? Notice that as we go from left to right, we're adding more and more carbons. And so this is the trend. We're going from most polar to least polar. Some people seem to want to tell me about acidity and things like that. That has nothing to do with these solvents. None of these solvents are acids. Okay, in fact, they are all polar solvents, but they're getting less polar as we go from water towards propanol. I'm showing you here, just in case you don't recall, we talked about fatty acids earlier on in the semester, and remember that they are very nonpolar. You can see all those carbons and hydrogens on there. And what we're seeing as a trend is getting more and more hydrophobic, less and less polar as we go along. And that's what I want you to be discussing in terms of what is going on with the solvent results. Okay, where are we now? All right, we are ready for topic 7. Like I said, I will talk about the lab reports a little bit more on Wednesday regarding citations, and then they're due on Thursday. So don't forget to get to that now and make sure you hand it in on time. It's worth quite a bit of your final grade. So topic 7 is all about the cytoskeleton and molecular motors. And it's a short topic, but I'm not sure if I'll quite finish it today. I've just used up 20 minutes looking at the midterm and stuff, but we'll see We'll see where we get with this. And I showed you this picture before. I was trying to show you this picture when we were talking about the cytoplasm to show you that the cytoplasm is not just this empty void full of water. There's actually quite a few things in it. But one of the major components that's in there are these protein fibers that are known as the cytoskeleton. So I'll show you some more pictures of the cytoskeleton. There's another picture. There's a lot of people who use fluorescence microscopy, and it's a very good technique for looking at the cytoskeleton. And yeah, lots of great pictures. You can see wonderful pictures here with fluorescence microscopy and lots of cytoskeleton showing up there. There's another one. You can see the nucleus quite clearly and mitochondria in these. I really like some of these pictures. So there's a definition of cytoskeleton. It says it's a proteinaceous. That means it's made out of proteins. And it's these fibers that are all over the place. And so what we want to do is talk about what they are doing. There's another little cartoon showing it integrated in there with the organelles and all that. And there's three types we're going to talk about. And this is the worst thing about the cytoskeleton are these three types in terms of the names. These names are awful because they just all sound alike if you're not familiar with them. Microfilaments, intermediate filaments, and microtubules. So sorry about that, I didn't name them. I'll try to help you get through it though with today's lecture. So the microtubules, this is a quick introduction, are the largest. The microfilaments are the smallest. And the intermediate filaments are intermediate, which means they're in the middle. So hopefully that will help you remember that they are in the middle. One other thing you might notice from these diagrams is there's a lot of cell photos that have this specific color because these are common stains. And so you'll see often that the nucleus is stained blue, the cytoplasm green, and the kind of cell surface stained orange or red. And again, that has a lot to do with the stains that are used, and particularly in the textbook. So something else to look out for is the colors. And notice that all of these microtubule components are different locations in the cell, and that's going to reflect their functions. So that's kind of an important concept to think about. So let's get into this. I see there's a question, somebody asking about what should be about the polar stuff. Take a look at the questions in the lab manual. And like I said, when you're talking about the solvents, I want you to talk about the polarity of the solvents. Okay, that's what I'm looking for in that particular question. Okay, so like I said, let's get into this. Three components, and we want to talk about them. The one place to start is what they're actually made of. Okay, so that's why they're classified as different things, because their components are very different. It's like talking about you know, if you think of we could have rope made out of plastic fibers. We could have rope that's made out of cotton. You know, it's sort of the same idea. Very different subcomponents that we want to talk about here. So the microtubules is where we are going to begin. And what I'm going to do, as I've done with some of the other units, is I'm going to, there it is, I'm going to start a note here. And what I'm going to do is I'll just start filling in a couple of things for the microtubules. And this is what my table is going to look like anyway. And so as we get through the unit, we're going to fill in this table. So let's talk about microtubules. So microtubules, these are all made out of proteins, by the way. And these are made out of a protein called tubulin. So microtubules are tubes. Okay, so they're actually hollow tubes, and they kind of look something like this, right? So there's actually an inside to the microtubules, and all those are my little tubulin proteins. So just remember that, okay? Tubulin, microtubules, it's implying a tube. Anytime you see IN on the end of something, IN means it's a protein of some sort, okay? So it's a protein tube is what it is. The relative size, this one is the largest. Okay, and that's all I'm going to fill out in this table for now. I'm going to come back to it, but I just want to show you this table, and like I said, mentioned we're going to talk about it. So notice the last two things, we've got functions and we've got cellular location. So those things are going to be important. Okay, what happened to the PowerPoint now? I think we have, is this it? Oh no. Back to the PowerPoint. Oh, there it is. Okay. So let's talk about microtubules. Okay. So microtubules is a fancy name for something that you may be familiar with already. If you've heard of spindle fibers. Spindle fibers is usually the name that we give to them when they are part of mitosis, by the way. When they're not part of mitosis, they're usually just called microtubules. Again, it's one of those things where, you know, in the area of biology, this is something that, you know, people in different fields sometimes have different names for the same thing. And it's just unfortunate, but that's the way things are. So microtubules are basically what centrioles are, what microtubules grow out of. So you can sort of think of a centriole as the roots or the seed of the microtubules. So notice in this diagram here, you can see if you look carefully, it looks like a whole bunch of little tubes. Okay, so we want to kind of talk about that in terms of, we'll come back to that in terms of it being one of the functions. So microtubules, by the way, are what make up flagella and cilia. So we talked about these particular organelles in lab 5. So last Thursday, we were looking at flagella and cilia. And flagella, of course, are found in sperm cells. And cilia are found in the paramecium. And I think I'm going to have some videos here. So there's a sperm. And so this is very different from the bacterial flagella. And that's something I want to talk about today, is how it is different. Remember, a bacterial flagella spins around, it rotates clockwise and counterclockwise. This is a bit more like a giant tail that whips back and forth. Cilia are very similar. They kind of whip back and forth, but kind of just on a smaller scale. In terms of the length, you're looking at, you can see that a flagellum is much, much bigger. Here's from some of the organisms we're looking in the lab. And you can see on the left, there's a euglena with a flagella. And if you look carefully at the video there, you can see that flagellum. I'm not sure how they did such a great job of looking at it. And the cilia on the paramecium, you can see, maybe you can see them on the surface. They kind of look like little blurry flickers. Kind of hard to see them, you know, with a light microscope in generally, but you can see they're stained here on the diagram on the left. So let's take a little bit of a closer look at these things. If you make a cross section of a cilia or a flagellum, it kind of looks like this. So if you take a look, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9. This is often called a 9+1 arrangement because there's 9 around the edge and there's 1 in the middle, and they're kind of actually like 2 tubes stuck together. Anyway, it's kind of an interesting little structure, and this slide is also reminding us that we do have cilia in the human body, and they are actually found in your respiratory system, and they capture mucus and they capture pathogens and they flush it out. And they do the same thing. They wiggle and they push mucus out of you. And most of the time you don't really notice it, except for when you're sick, you make a lot more mucus. And so that's the time that you're going to notice it. Here's another electron micrograph. You can see that arrangement there of the cilia and all the 9+2 arrangements. And you can see on the right is a cartoon. And you may notice from this cartoon that this whole thing is surrounded by a membrane. So that's a feature I want to point out here, and I want to talk about that and come back to that in a moment. I think I have a couple more pictures to show you here. So this is showing the arrangement of a flagellum here. The other thing to notice is right here, these little red things here, those are motor proteins. So it's important to know what a motor protein is and what these things are doing. So if you take a look at this structure of a flagellum, you've got these microtubules, right? And I'm not going to draw very fancy microtubules, but they're these big tubes. And what we want to do is we want to swim. So our body has installed these little dynein proteins, and I kind of think of them as like this, as maybe a couple of legs. And what they can do is they can flex those 2 tubes back and forth, and that's going to make the whole thing flat back and forth. And the other key thing to know about motor proteins is they take energy. take energy in the form of ATP. So I think I've got another slide here showing the movement of the microtubules, and you can see the ATP is being hydrolyzed over here, and this is causing the bending action to happen here, and in the end we get a wave-like motion of these flagella. One other thing to know is that there are motor proteins all over the place. And we talked a little bit about organelles moving around a cell, like vesicles, and it's really just the same idea, motor proteins again. So I'm going to come back to that in a second. But what I want to do is stop now, and I want to draw something for you. So where is it? Here we go. So I'm going to make some drawings for you. And what I want to do is talk a little bit more about the bacterial flagellum and the eukaryotic flagellum. So I'm going to draw a picture for you. So you're going to need probably a full piece of paper, and I'm going to label it. So go back in your brain. You're talking about the bacterial flagellum in topic 5. And what I want to do is draw a very, very rough picture of a bacterial flagellum. So here goes. So what I'm going to do is I'm going to draw something here called a basal body. And so this is basically a bunch of proteins that anchor this thing into the membrane. So what we need to do is draw the membrane too. So let's draw the membrane. Here we go. And let's say this is E. coli. So E. coli is going to have 2 membranes, and it's going to have a peptidoglycan layer. So let's label that here. Okay, so we have our plasma membrane. We have our peptidoglycan. And like I said, in E. coli, there's an outer membrane as well. Remember, it's a gram-negative organism, okay? Okay, so that's the first part. Now, the next thing we're going to do is draw something called a hook. And okay, so let me try that again. Let me try that again. Like I said, I'm not the fanciest artist, but I do want to draw this nice. There we go. So that's a little better, I guess. So this is called a hook. Again, this is made out of protein. This whole thing is just a bunch of proteins put together. Now the basal body, you can almost think of it as a molecular motor. So maybe I will put a note on that. And we'll just call it, I'll label that right now as the basal body anchors into the membrane or the cell wall and has a molecular motor. So it's a tiny little motors in there, which is going to allow this whole thing to spin around. Remember, this whole thing spins around? Okay, so let's draw the rest of it. So the rest of it is very long, and I'm going to just draw it way over here. It's probably longer than what I can fit on my screen. And that is, I guess you could call it the tail. And so I'll label that as the tail. And this is made of something called flagellin protein. So made of, if I zoom in, I can write a little neater. Flagellin proteins. And that's my drawing of my bacterial flagellum. Now I'm not quite done yet. There's a couple other notes we need to make around this, okay? And maybe I'll do the other notes in some other colors. One thing to note is remember that the motion, okay, so the motion is basically rotation. So it can move clockwise and counterclockwise. And the other thing is to talk about how it's powered. And this is powered by something called the proton motive force. Now, I have not talked about the proton motive force yet. We're going to talk about it when we talk about respiration. but just know we're going to come back to that. Okay, so that is my bacterial flagellum. So maybe I'll just move the title over here, and then that way it looks a little neater. And if you don't get all this, you can always ask me for it anytime, or you can watch the lecture recording. So that's the bacterial flagella. So like I said, you know, you're talking about something that's in the membrane, and it sticks out and it rotates around. So let's draw the eukaryotic flagellum now for comparison. So it's not going to be, again, too fancy, but I will do my best to draw it for you, and then we'll label what I'm looking at. So it kind of looks like this. Okay, I'll come back to and you'll see what I'm trying to draw. There's sort of a weird fold anyway, and that's what I'm trying to show you is the weird fold. Okay, so there we go. And maybe what I need is another color for the microtubules. I'm not going to draw, there is something called a basal body. Maybe I will draw it in another color here. And it's very, very different from the basal body of the bacterial flagellum. And it's just basically shares the same name. And so maybe I will draw a note here. So this anchors in the membrane. And you're probably wondering, what on earth are these squiggly lines here? These squiggly lines are the membrane. So let's label that as well. So what I'm trying to show you is that the membrane goes along and then it folds over and then it surrounds the entire flagellum. Okay. All right. So sorry for all the moving around. It's kind of hard to write on the screen. I got to zoom in to often write. Okay, so this basal body, the microtubules now grow out of this. Now, I'm not going to draw 9+2 microtubules. I don't know if I'm capable of drawing that very neatly. But what I am going to do is draw some microtubules. There's some microtubules. I know they look very beautiful. I'm going to draw 2 sets of microtubules, and that's it. I'm not going to draw a 9+2. Uh-oh, what just happened on that? There we go. Okay, so let's label that as microtubules. Microtubules. Usually I do this on a whiteboard so you can see the whole picture all at once. So I'll just zoom out for you so you can see everything there. Okay, so there's my microtubules. What are we missing? We are missing our motor proteins. So I'll draw those now for you. So here's some motor proteins. I'll draw a few here. They, like I said, they kind of look like this. Maybe I'll draw one more for you. Okay, and so we can label this as dynein. Motor proteins. All right, so the last 2 notes are basically the same last 2 notes that we had for this other one. What does the motion look like, and how are these things powered? So the dynein motor proteins, remember, these are powered by ATP. So we can write that down. Powered by ATP. Technically ATP hydrolysis, but we'll just say ATP. And the last one is the motion. And the motion, maybe I'll squeeze it in over here. Okay, and the motion, come on, there we go. Motion is back and forth. Back and forth. Okay, so not the most amazing diagrams, but there's a number of points that I'm trying to make. One is that the structure is completely different. They're made of very different proteins. 2, the eukaryotic flagella is surrounded by membrane as well. 3, they have different motion. 4, they're powered differently. Maybe the last note to make is the size. So we can write that down maybe here on the bottom. Size of the Bacterial Flagellum. So Bacterial Flagellum is maybe 1 to 5 micrometers, kind of is the typical range. And the Eukaryotic Flagellum, we're looking at much, much larger cells. So in this case here, we're looking at, depending if it's a cilia or flagella, you know, cilia can be sort of in the smaller range, maybe about 10 micrometers, but we can be up to maybe 200 micrometers for some flagella. And so much, much larger structure. Very different structures, even if they have the exact same name. Okay, time for a time check. All right, let's get back to microtubules. And maybe what I'll do before I get back to the PowerPoint slide is I will go over to our Word document and write down a couple of the things that we've talked about. The cytolocation is basically the cytoplasm. And functions, we have cilia and flagella. We can write that down. And we also have, we talked about organelle movement. So we're not quite done with microtubules. So I'll come right back to this in a moment, but I just wanted to fill in a couple of details I was thinking about. Oh, that's not the PowerPoint. That is the PowerPoint. Okay, so this PowerPoint here was just talking about flagella movement and the motor proteins, and this is showing more motor proteins in action and those vesicles moving along the microtubule. You can kind of think of it as a little monorail or train track or something like that, right? And there's my reminder to do the diagram on the board, and I guess I just did that already. You can see this is something I popped off the internet. You can see they're emphasizing different structures and different motions as well. So I want to show you this video here. I'm not going to press play quite yet. This is some sperm cells, and what they're going to do is they're going to trim a flagellum off. And so watch this, it's pretty cool. So they trim it off, and notice it's still moving. So this is because there's still ATP within that membrane. If this was a bacterial flagellum and they trimmed it off, it would just stop dead. The power source is at the membrane, whereas in this case, the power source is the ATP, which is in the cytoplasm, and the cytoplasm is in throughout the entire flagellum. So pretty cool. I'll just show you that one more time. I think it's a pretty cool video. There we go. Oh, yes, back to the Word document here. Where's the Word document? I guess what I forgot to do is put down the other thing that we talked about is, of course, spindle fibers. And we're going to be talking a lot about spindle fibers when we talk about mitosis. And so we'll come back to them and talk about them in a lot more detail. So there's a question here. It says, why are they cytoplasmic if they're attached to the membrane? They're not attached to the membrane, they're inside the membrane is what they are. And that's, of course, by definition, cytoplasmic. So that's kind of a bit of the microtubules. Remember, they're made out of tubulin. They're the biggest ones. They're in the cytoplasm, so they're moving around organelles. They're involved in mitosis and meiosis. And of course, the other feature is the flagella, which is something we've talked about. So let's see here. We've got about 5 minutes left. Let me just take a quick look at the notes to see if it's worth it moving on, because I might have to just say, hey, that's enough for today. Uh-oh, what's it doing now? The computer is not happy with me. So hold on a second here. So I'm just looking ahead, and I think it's not worth it to talk about the next part. The next part is the actin filaments, and it's going to take us more than 5 minutes to really talk about actin filaments. So I think what I will do is maybe finish the lecture off there today early, and we'll come back and we'll just have to finish up topic 7 next day and get into the following one where we're going to talk about energy.